March 24th. A young German physician working in complete isolation from the scientific community of his day went to Berlin and gave a lecture to a very small group of other physicians. One of the physicians that was there went home and later recollected that evening lecture, and I quote from his journal entry. "I hold that evening to be the most important experience of my scientific life." The name of the physician that gave the lecture was Robert Koch, who went on to win the Nobel Prize in medicine and completely change our lives. Many of us, perhaps most of us, are only alive today because of what that man did. The physician who attended the lecture and wrote in his journal's name was Paul Ehrlich, who later went on to win the Nobel Prize in medicine and became universally recognized as the father of immunology. Today, the great-great-grandnephew of Paul Ehrlich, Garth Ehrlich, is here today to turn the world of Robert Koch upside down. So I hope the historical irony of today's lecture is not lost on anyone. I've asked Garth to come here and give a relatively rigorous explanation of his distributed genome hypothesis, because our specialty, the entire edifice of endodontics, is based upon what Robert Koch did. And everything that we do goes back to that. And if that edifice is under attack, then as endodontists, I think we better know something about it. So I've asked him to come and give this explanation of how our world is about to be turned upside down, and if nothing else, we will confirm the old saw that the apple does not fall far from the tree. Garth. I would have reprinted his CV, but it's 92 pages long. He's one of the most accomplished scientists I've ever met in my entire life, so I know you're going to really enjoy his presentation.
Gary, thank you for that incredible introduction. So we heard from Dr. Bate about the importance of mutation and the difference between life which replicates with mutation versus crystals which replicate perfectly. And so what I'm going to be talking about today is the importance of mutational mechanisms in the evolution of bacteria. And what I want to, if you leave with nothing else, is to understand that during the chronic infectious process, one of the things that allows for persistence over time in a patient is the fact that the bacteria are actually changing during the infection. And so it's their ability to mutate during the infectious process which leads to persistence. So I just want to start by thanking a fairly large number of people who have done the work that I'm going to be talking about today. And we're going to be going over a lot of DNA sequence information And this all really came from Fen Hu, who set up this system in our laboratory, and her team of sequencers and bioinformaticians. And the clinical perspective from all this came from my longtime clinical partner, Chris Post. And I want to also acknowledge Dr. Bill Costerton for inspiration, because if I hadn't come across his work about 15 years or so ago, I would have never come up with the work that I'm going to talk about today. So let me just start with some simple definitions that we use so that everybody's on the same page. Genetic heterogeneity means that there are different alleles of the same gene present among individuals of a population or a species, and this is what most of us are familiar with who had a basic course in genetics at some point. What I'm going to be talking about more today than genetic heterogeneity is what we call genomic plasticity. And this is very different. It says that there are different genes present among individuals of a population or a species, such that all individuals of the same species do not have the same set of genes. Now this, as we go through the lecture today, we'll find that this is pretty much universal for prokaryotic organisms, at least for bacteria. We haven't looked that much at archaea yet. And it may apply to some asexual eukaryotes. But any organisms that go through sexual reproduction you're not going to find this type of genomic plasticity. And so as Gary was saying, we as microbiologists, and particularly microbiologists who study infectious disease, we all owe an enormous debt to Robert Koch. And his paradigms were developed to explain what were the prevailing infectious diseases of the time. These were acute clonal epidemic diseases that would run through populations and kill very large segments of the population. And the system that he came up with very accurately modeled what was going on in these acute infections. And so the idea, of isolating, a single bacterial isolate, putting it in culture, taking it out, putting it in an experimental animal, showing it caused the same disease, and then re-isolating it, was a very valid and paradigm, and it still is if you're dealing with acute clonal infections. What I really want to stress today is that there is a fundamental difference between acute infections and chronic infections, and Koch's postulates don't apply to chronic infections. So there's this very important dichotomy between chronic and acute infections. And you'll see me over and over again in the talk today. I'll be referring to some of our work where we study middle ear disease or otitis media. And this is a very useful dichotomous model because there's an acute form of otitis media and a chronic form of otitis media. And recently I have learned from talking with Gary and his colleagues that this is not unlike what you see in dentistry, that you also have this dichotomy of acute and chronic disease. And so some of the differences between chronic and acute infections, kind of the yin and the yang, if you will, of microbial infections, is that chronic infections are associated with biofilms and acute clonal infections are associated with planktonic phenotype. Now, I'm not going to talk too much about biofilms because after lunch you're going to have a lecture from the world's expert in this area, Bill Costerton. So I'm going to keep my comments to biofilms fairly limited. Another major difference between chronic infections and acute infections is that chronic infections tend to be, in most cases, polyclonal, whereas acute infections are almost always monoclonal. So by polyclonal, I mean that you have multiple strains of the same species present at the same time. And you'll find out later on why this is so important. Chronic infections can also be polymicrobial versus single species, that is multiple species, but they don't have to be. And the reason that polyclonality and perhaps polymicrobiality, if that's a word, are important is because it's this ability to generate diversity through horizontal gene transfer among the strains and between the species that are present in these chronic infections. And so that if you have horizontal gene transfer going on between the strains or between the species, you're constantly having mutation, you're constantly generating new strains, and some of those strains will have a selective advantage in the host. And so they're essentially staying ahead of the host, if you will. The host has an immune system, and then our immune system works by gene rearrangements, if you will, by mutation. And so this horizontal gene transfer for the bacteria acts as a counterpoint, if you will, to the host's adaptive immune response. And what I want to stress is that these acute-chronic dichotomies apply across all medical and dental disciplines. We started our work in otolaryngology looking at middle ear disease. We've now shown that the same thing works for adenoiditis. This is a picture of an adenoid. And every one of these colors, this is a fish, a fluorescence in situ hybridization. And every one of these colors on here represents a different bacterial species. So you can see that this is this is polymicrobial. You have many different species here colonizing or infecting these adenoids. Same for chronic sinusitis, cholesteatoma, tonsillitis. You go into urology and gynecology, prostatitis, endometritis, cystitis, vaginitis. These are chronic conditions in general. And in orthopedics, osteomyelitis, infection of artificial joints, arthroplastic infections, and in dentistry you have periodontitis, and as I learned today, apical periodontitis. And so we believe that all of these conditions can occur as both acute and chronic forms. Now, biofilms are what we call a population-level virulence factor. And what I mean by a population-level virulence factor is that it's a disease-associated mechanism that exists or occurs only at the population level, in this case for the bacteria. So that these are traits that are not of individual bacteria, but rather are associated with multicellularity. And so this, I think, is a very important concept, that when we're dealing with these chronic infections, you have to think of the bacteria truly as multicellular organisms, and the individual bacterial cells are no different from the individual cells that we have in our tissues. There's differentiation in terms of genotype, phenotype, and metabolism. And in general, these population-level virulence factors, again, are associated with chronicity or persistence, as opposed to acute virulence traits such as toxin production. Most people think of virulence traits in bacteria, they think of things like toxin production or adhesions which allow the bacteria, an individual cell, to stick or to acquire heme or iron from the host. And these are very different from population-level virulence traits, which are more associated with chronicity. So, biofilms have an extracellular matrix, which you can think of as analogous to the extracellular matrix that you have in your tissues, perhaps, in any tissue, whether it's a bony tissue or a soft tissue. And one of the major components of that matrix, believe it or not, is actually DNA, extracellular DNA. We refer to it as eDNA. And this is true for all the bacterial biofilms that have been examined, whether we're talking about Pseudomonas aeruginosa, which is associated with the CF lung, Haemophilus influenza, and Streptococcus pneumoniae, which are associated with otitis media and other respiratory tract infections, or Enterococcus faecalis, or Streptococcus mutans, which of course is associated with dental caries. They all have DNA as a major constituent of their biofilm matrix. And so this is a picture from a paper that was published in the Journal of Bacteriology by our colleagues Jurecic and Bakalitz. And the blue, represents strands of DNA in the bacterial biofilm. So you can see that it's a very major component of the bacterial biofilm. And if you look, this is just a gel where we were looking to see if there is extracellular DNA in cultures of Haemophilus influenza. And so if we look at a planktonic culture, which is here in this lane, course, we see no DNA at all. So the DNA being expressed is not something which occurs when they're growing acutely or in planktonically. Obviously, if we lyse them, then we can see DNA. This is a planktonic culture lysed. But this is the biofilm culture here, unlysed. And you can see there's massive amounts of DNA, extracellular DNA here. We also showed indirectly that the pneumococcal biofilm DNA as a very major component. So this is a 4-day-old pneumococcal biofilm here, nice robust biofilm structure. We treated it for 15 minutes with recombinant human DNase and debulked it by 90%. So again, you can see that there's DNA in the biofilms. And later on, we'll talk about why it's so important to have this DNA there in the biofilm as an extracellular source. What I want to stress is that biofilms are really the most important of the population-level virulence factors because they potentiate other population-level virulence factors, including what I'm going to be talking about today, horizontal gene transfer. Okay? Because without the biofilm, you don't have a good medium to support the horizontal gene transfer. Biofilm brings the bacteria in close proximity. It supports polyclonal colonizations, as we talked about before. It supports polymicrobial colonizations. And it provides a stable environment for mating. Bacteria can mate. This is some species can, at least. This is a pseudosexual process. It also provides a matrix for maintaining DNA in close proximity to bacterial cells for competence and transformation. So these are two different mechanisms, mating and competence and transformation, that bacteria use for horizontal gene transfer. And they're mechanistically somewhat different, but they both end up in with the same result, that you're transferring DNA from one mature cell to another. So if this is the biofilm matrix and you have a couple of different cells here, the matrix provides for a stable relationship between these cells so that you can have DNA pass from one into the other, and then this second cell becomes transformed, making now a third type of cell. It's been shown in numerous other laboratories, a lot of this work has actually been done in Denmark, that biofilm growing cells experience transformation rates at up to 10 to the 4th fold higher than planktonic cells. That's 10,000 times the rate. An analysis of multiple complete bacterial genomes from more than 24 bacterial species reveals that 5 to 40% of all the genes in all the genomes of all the species we looked at are horizontally acquired. That is, not acquired, from mother to daughter cell, but between mature cells. So why do we care so much if bacteria undergo horizontal gene transfer? Well, working with clinicians, one of the things that I was struck by was that individual clinical isolates of both Haemophilus influenza and Streptococcus pneumoniae, which are a couple of my favorite pets, display profound differences in phenotype and in the virulence they produce and the mortality that they produce, both in humans and in experimental animal model systems. So if you have these profound differences in phenotype, it suggests that there's probably profound differences in genotype underlying that. And so here's just an example of some differences in the biofilm phenotype of 6 different clinical isolates of Streptococcus pneumoniae, the pneumococcus. And so what you can see here is we just simply grew the pneumococcus on in glass flow cells, and they did confocal microscopic imaging. And so you can see that some of them, you'll get just a little bit of sticking to the surface, and other ones grow very lush, robust biofilms. So there's very large differences, very easily apparent in the phenotypes of these different strains of the same species. To make the point even more so, these are 3 different strains of pneumococcus isolated from the same patient. So not only do you see differences among strains from different patients is, but you see profound differences within a patient as well. And then if we look at the amount of disease that these different strains cause within an animal model, and the animal model we use here is a chinchilla, and it's a middle ear infectious model, and we have a quantitative score for virulence here on the x-axis, and then these are just different strains of Haemophilus influenza here. So we can see some of them cause very little in the way of disease or virulence, and other ones cause intermediate levels, and some cause very high levels of disease. Similarly, if we look at these same strains and look at the amount of mortality that they show after we do our experimental infections, some of them cause no mortality at all, some cause low levels of mortality, some intermediate, and this strain kills 100% of the animals that we use. And so we'd been accumulating this data over a fair amount of time before we actually got into the genomics of the situation. And we can see the same thing with the pneumococcus. I won't belabor the point, but you can see there's very large differences in the amount of disease the pneumococcus, different strains of pneumococcus causes, and again, very large differences in the amount of mortality they cause in the animal models. So we surmised, based on these extensive phenotypic differences among the clinical strains, there must be a great deal of genetic heterogeneity among these strains. This was before we had the concept of both genetic heterogeneity and genomic plasticity. So it's implicit in the biofilm paradigm that there exists significant phenotypic heterogeneity among genetically identical envirovars because we know the same strains can form biofilms and grow planktonically. And this has proved to be a particularly useful rubric for informing us with respect to the study of chronic bacterial infections. It is, however, incomplete as a model of chronic bacterial pathogenesis. And so we came up with this concept of bacterial plurality, which includes the biofilm planktonic dichotomy, but it embodies the concept that bacteria within a population display heterogeneity at many levels, not just the phenotypic state as we've been talking about. Of course, here you can see a picture of actually a biofilm bacteria growing on the middle ear mucosa of a chinchilla, but the differentiated function. And this is a picture from Phil Stewart at Montana State University, where you can see that this is a biofilm here, and the green represents metabolically active bacteria on the surface of the biofilm, and the red represents metabolic quiescence in the interior of the biofilm. So we know that they're differentiated with regard to metabolic function as well as phenotypic state. And what I'm going to talk about for the rest of the talk is the differentiation with respect to gene content. So I've hinted at this. What are the origins of genomic plasticity? Well, pharyngeal carriage, whether you're studying periodontitis or whether you're studying otitis media, is polyclonal for almost any of these pathogenic species. Chronic infections in carriage are associated with the biofilm phenotype, which we already know is a population-level virulence factor. We've seen that the biofilm matrix contains bacterial DNA. We've also observed in vitro that gene exchange is elevated in biofilms, and that most chronic pathogens have energetic systems for acquiring DNA. This was one of the things that really struck me about 10 years ago when we just started to think about this. When we're all microbiology students, we all learned about transformation. We know that's actually how DNA was first discovered as the hereditary substance from the experiments of Griffith in 28, where you could transform live pneumococcus into non-virulent ones into virulent ones using some substance from dead virulent bacteria, and then that was shown 16 years later by Avery and colleagues at the Rockefeller to be DNA. And so these experiments were extremely important from a basic science point of view, but it took another several score years, I think, before we realized the importance of this from a clinical infectious disease point of view. And one of the things I want to emphasize is that most of these pathogenic bacteria have what are called reduced genomes. They have small genomes that have, between 1.5 and 2.5 megabase genomes, whereas most bacteria you find living environmentally have genomes of 2 to 3 to 4 times that size. So there's obviously a fair amount of pressure on these bacteria to keep their genomes small. Yet in spite of the fact that they have small genomes, they devote a fairly extensive percentage of their genome to mechanisms for bringing other genes into their genome. Some of them have multiple mechanisms for doing this. So, for example, if you look like at Haemophilus influenza, 2 to 3% of its genome is associated with bringing in genes from foreign sources. So pathogens evolve during colonization using horizontal gene transfer mechanisms, which result in the continual production of new strains. So what's some of the evidence that chronic infections, we've already talked about chronic infections are associated with the biofilm phenotype. Nasopharyngeal carriage in chronic infection is polyclonal. Now this, of course, is the challenge to Koch's postulates that chronic infections bring. And when I first proposed some of the work that we're doing, I was very fortunate. In the first time that I talked about it publicly, Tim Murphy from the University of Buffalo was there in the audience, and he had evidence back in 1999 that every one of his patients with chronic obstructive pulmonary disease, what I'll call COPD, had multiple strains of Haemophilus influenza present at the same time that he could tell by pulse field gel electrophoresis. More recently, Janet Gilsdorf at the University of Michigan has shown that Haemophilus influenza carriage in the nasopharynx is always polyclonal. She's shown that it can be as high as 2 or 3 dozen strains present simultaneously in the nasopharynx. Similar work has been done for pneumococcal carriage and infection by Dowson in England and De La Castri in Portugal. And more recently, our group has shown that, in fact, not only in colonization, but also in infection. And then the Danish groups, Molen and Hoiby, have shown that you have polyclonal infection in the CF lung with Pseudomonas aeruginosa. So this is, it's not one species, it's not one condition. It's every place he's looked, we find polyclonal colonizations and polyclonal infections. So as I was alluding to before, pathogens evolve using highly efficient HGT systems which generate unprecedented levels of genomic plasticity. And so these are just the different mechanisms for horizontal gene transfer that different species use. Haemophilus influenza has been known for years to use competence in transformation, but about 3 years ago it was shown that many strains can also mate. Moraxella cataralis uses competence in transformation. Pseudomonas aeruginosa mates. Pneumococcus, like Haemophilus, was known to be competent. Again, it's recently shown that it can also mate. Staph aureus uses phase-mediated transduction and mating. Strep mutans uses competence in transformation. So we don't find any of the pathogens that don't have energetic mechanisms for the take-up of DNA into their systems. So this brings us to the distributed genome hypothesis. And the distributed genome hypothesis says that chronic bacterial pathogens utilize a survival strategy wherein a majority of their genes are distributed among a population and are not found in all members of the species. Thus, at the population level, there exists a supergenome which is much greater in size than the genome of any one organism for that species. And the virulence corollary to the distributed genome hypothesis is that this distribution of genes among a population again serves as a supervirulence factor or a population-level virulence factor, just like a biofilm, that provides for improved population survival through continual horizontal gene transfer mechanisms which provide for rapid adaptation to environmental conditions through the reassortment of genes. And obviously, alleles reassort as well. And so this is just kind of a cartoon that we've developed to kind of explain the process. So if each of these little ovals down here represents a bacterial strain, and this green helix represents the core genes for this. These are all of the same species. This green helix here represents the core genes, or those genes that are shared by every member of that species. But there's also these population level genes which are different among species, and so each strain gets a different assortment of genes from the population level supergenome. And if one of the bacteria dies in a population, then those genes go back into the environment and can be picked up by one of the still existing strains, making yet another strain. So we had this observation that each strain or isolate within a species has its own unique set of strains derived from a population level supergenome. We have the problem, how do you characterize this genomic plasticity and determine relationships among strains or species? Because this was done by sampling incomplete genomes. When we started this 10 years ago, we didn't have the capability to do whole genome sequencing at that time. So with these thoughts in mind, we developed a strategy to comparatively analyze the genomic characters of large numbers of strains of several pathogens. Because when we first proposed this about 10 years ago, even we ourselves really weren't sure what we were going to find. So we chose a group of test organisms: Haemophilus influenza, Streptococcus pneumoniae, Staph aureus, Pseudomonas aeruginosa, and Gardnerella vaginalis. And these three organisms collectively cover gram-positives, gram-negatives, gram-indeterminates, what we call professional pathogens, that is, organisms that live only in the human body, and opportunistic pathogens, those that can live elsewhere as well. Because we wanted to have representative species for all these different classifications. And so from our initial studies, where we were just doing sequencing of standard, what are called genomic libraries, made from multiple strains, what we found is that no matter how deep into the libraries we sequenced, and we weren't doing whole genome sequencing, that greater than 10% of all the genes that we sequenced from Haemophilus influenza, the pneumococcus, and Pseudomonas aeruginosa were unique. No one had ever seen these genes before. These were published in a series of 3 papers by Fen and her postdoc, Kai Shen, in Infection and Immunity in the middle of the last decade. And what we found was that these new genes that we were finding weren't confined to any one class, okay? They fell within all possible functional groups, okay? You'd find adhesins, you'd find sugar metabolism, you'd find toxin production, you'd find endopeptidases. It was all across the entire spectrum. So it just seemed to be a very broad universal phenomenon. When we were looking at Haemophilus influenza, we found that the distributed genes show amino acid homology to proteins from other species, but mostly related species from within the Pasteurellinaceae, which Haemophilus influenza is a member. So we had enough evidence in 2003, 2004, by doing this kind of random shotgun sequencing, that we were pretty sure the distributed genome hypothesis was correct. But what we really wanted to do was be able to look at whole genomes of large numbers of organisms. And fortunately at that time, I ran into Sean Lonergan, who was one of the guys who was developing the 454 sequencer, and we started a collaboration with them. And Fen actually went out to their site there in Branford, Connecticut, and did large-scale sequencing as an alpha tester back in '84, and then we were one of their beta test sites in '85, and since that time we've sequenced somewhere over 150 complete bacterial genomes. And the 454 sequencing system is unique among the high-throughput DNA sequencing systems in that the read lengths are long enough so you can actually assemble de novo whole bacterial genomes. None of the other systems out there currently can do that. Because the 454 is so important because the heterogeneity among strains of a species is so vast that resequencing, you can never rebuild a genome if you have what are called short reads. And so this just shows the sequencing that we've done at our center. These are graduate student Ben Janto and Azad Ahmed, a postdoc, who have done the great bulk of our sequencing. This is just looking at how much sequencing we've done for these two species compared to the rest of the world using this 454 technology. And so this is just as my colleague Chris Post, a surgeon, says, he says, oh God, not knowing your PhD slides, Garth. But this is just to show that these are the strains of Haemophilus influenza that we sequenced. We started out just looking at strains from Children's Hospital in Pittsburgh, but then we branched out and looked at other regions of the USA, Canada, Malaysia, Papua New Guinea, and across the US. And what I'll tell you is that we see as much diversity in the strains within Pittsburgh as we see over the whole world, so that there is diversity at every level. Now, when you start sequencing large numbers of bacterial genomes and you want to start comparing them, you have to develop the bioinformatics to actually do these comparisons. And I'm not going to spend a large amount of time talking about that, but we really had to develop our own tools because when we started there just was nothing out there. And I want to, whoops, somehow I lost one here. All right, so I lost it. What I want to do is acknowledge Justin Hoag, who was a polymath, who was in our group at the time, who really developed the computational structure for doing this. I would ask him a question, and each day he would come back to me the next day with a fully formed and functioning system to do the analysis. And so some of the questions that we wanted to talk about with regard to comparative genomics are largely questions related to genic content or gene content. And the most important thing before we do anything else is how do you cluster genes to ensure that orthologs are not counted as different genes? That is, you don't want to, if you're binning your genes and you want to say, okay, this is, a new gene and this is an old gene. You don't want to take a gene which is very closely related to another gene, probably does exactly the same thing, it's just an allelic difference, and say, okay, it's a new gene. So you have to have a system that allows you to truly bin what are called orthologs from paralogs. Paralogs are genes that have similar structure but different function, and orthologs have the same structure and the same function. Because we needed that data in order to be able to determine what the core genome size was for a species, what the supergenome size was for the species, and what the average number of genic differences were between any two clinical strains. There it is. Okay. I thought it was on the last slide. So this is Justin Hoag, the fellow that developed the pipeline. And so this is the most technical of I think of my slides. This is Justin's gene clustering algorithm, and we use what's called a single linkage algorithm because we were dealing with incomplete genomes. We were taking the raw data as we were getting it, so that we didn't have a single closed loop. We had the entire genome. But we hadn't put them all together because that takes a lot of time. And so a single linkage algorithm will cluster as one gene two or more sequences from a strain that both have homology with a single longer sequence from another strain. And this is very useful when you're working with unfinished genomes. And what we found is that linkage conditions were empirically determined by examining the conditions that minimize the rate of change in the number of clusters, which was identified as an inflection point when graphing the number of clusters versus the percent identity. And so that's what's on this next slide here. You can see if this is our identity threshold on the x-axis and the number of clusters we're seeing on the y-axis, what we see is that there's an inflection point. It doesn't matter what our match length is, right here at about 0.7. And so what's on this side tend to be orthologs, and what's on this side tend to be paralogs. And we actually experimentally verified this with a fairly large number of genes. And this inflection point occurs because of the fact that there is a fairly sharp break between what are paralogous and orthologous structures at this level of identity. So I've kind of gone over that. So when we're doing our sequencing, we would do our genome sequencing assembly as well as take publicly available genomes. We would then do our gene predictions, and then we would use Justin's algorithm for clustering orthologous genes. And so we would then bin the genes as either core, that was present in all strains, or distributed, means that they were present in only a subset of the strains of that species. And so this is just looking at the genomic statistics of all the strains of Haemophilus influenza. And one of the things that should immediately strike you is that if it's all one species, you'd expect them all to have the same genome based on genome size based on classic criteria. So what I've done here is I've highlighted the smallest genome in this group. This is 1.765 megabases, if you will, and this one is 1.984 megabases. So this one is 12% larger than this one. This would be like one human being having a 24th chromosome that was 50% larger than chromosome 1 that the rest of us didn't have. And so then we can also look at the number of genes or orthologous clusters that each strain has. And so we can see that this strain here has about 300 more genes than this one does. And if we look here at the percentage of what are non-core clusters, or the percentage of the genome that is non-core on a genic basis, we see the smallest genome still has 16% of its genome made up of distributed genes, and this larger one has 28% of its genome made up of distributed genes. We also look at the number of unique genes per strain, that is, genes that are not found in any other strain that's been sequenced. And so you can see that this strain here has 68 genes which are not found in any other Haemophilus influenza strain that's been sequenced. This small one here doesn't have any, but they range, you know, from 0 to 68 with pretty much everything in between. And of course, the more strains you sequence, of course, this number starts to drop. But what we really wanted to do was to model the size of the supergenome for the species. And so what Justin did was he took the number of core genes, which is on this plot here, and of course as you sequence more strains, the core decreases, but it very rapidly starts to level out. And this is the supergenome here. Of course you can see this is growing very rapidly at this point. And when we had data from 8 strains, he took this to develop a model to see if he could predict how many more genes we'd find in the supergenome as we continued to sequence more strains and what would happen to the size of the core genome. So the lines here are his model's prediction, and the triangles and the squares are the actual data. So you can see his model perfectly fits the data that we got as we added additional strains here. And this is his model out to 30 strains. And I just want to point out here, if you look like at about 17 strains, we're looking here at somewhere around, I think, 2,500 total genes. And if you look at 24 strains, you're looking at about 3,000 genes. So when we sequence 17 strains, here we are right on track with what his model predicted. And when we got out to 24 strains, we're right on track with what his model predicted. So his model is very, very accurate. And we've shown with our most recent sequencing, where we're working with Moraxella cataralis, that even with as few as 5 strains, it can predict the size of the supergenome very, very accurately as to what's going to happen in the future. So for Haemophilus influenza, what we found was that each strain differs at approximately 18% of its loci with respect to every other strain. So this has profound implications for what we were talking about before: strain differences with respect to disease phenotype, pathogenicity. We also calculated a number of other phenomenon for strain pairs, doing pairwise comparison. We did a similarity score, the total number of gene clusters shared between any 2 pairs of strains on average, the difference score, the number of gene clusters not shared, unique share, that is the number of gene clusters shared only within that pairing, and then a comparison score, which is a hybrid of the similarity and difference scores. And so this is just the analysis. And what I want to show you is that if you take any 2 strains of Haemophilus, on average, clinical strains, they will vary by 378 gene possessions. That's the bottom line from this. And their genome size is about, you've seen, between 1,700 and 2,000 genes. So this is a very, very substantial percentage difference between any 2 strains. And so at the point we are now, already having sequenced 25 strains, the core genome is less than half the size of the species genome. It makes up 47%, and the distributed genes make up 53%. I'll skip over this. And this is just a dendrogram showing the amount of gene sharing between strains. This is the number of gene differences on this axis, and these are the various strains here. So some you can see are fairly close together. They vary only perhaps by 100 to 150 genes, whereas some differ by 400 and 500 genes. And so if you look around the genome, you would expect, well, if there's all these differences, that there's going to be, you won't have synteny around the genome. That is, the genome of every organism is not going to have genes in the same order. And in fact, that's what we see. I just have you just pay attention to this last column here. What we're doing is we're comparing each of 12 clinical strains to a reference strain. And on average, there are 127 DNA insertions around the genome and 148 deletions around the genome compared to the reference strain. So that in total, there's about 275 insertions or deletions around the genome between every strain and every other strain. So what's the evolutionary origin of these distributed genes? Well, I had another postdoc in the lab, John Gladitz, who developed this least squares fitting routine to look at the origin of these distributed genes that we were finding. And I'm not going to spend time going through the details of how it works. We'll just go to the results. And what we find that is about 65% of the distributed genes for Haemophilus influenza had been evolving with the core genes of Haemophilus influenza for as long as the core genes. So by their codon usage characteristics, they look for all the world like the core Haemophilus influenza genes. They're just not possessed in every strain. And about 35%, or a third of them, come from other species, but most from related species. And what was interesting is that some of the well-known virulence genes are actually ones that have been introduced into the Haemophilus influenza supergenome. That was quite a surprise to us. Now, we did the same type of analysis with pneumococcus, a gram-positive, and I just point out here the percentage of each genome that is non-core, and it's even higher for pneumococcus than it is for Haemophilus. Here, even the smallest genomes have over 20% of their genome being non-core, and the largest ones have a third of their genome being non-core. And this is just, again, graphs of the core and supergenomes of the pneumococcus as we sequence more strains. And this work was published in Journal of Bacteriology by Fenn and our postdoc Louisa Hiller. And we found that on average, the pneumococcus differed at even more gene possession sites than Haemophilus, on 400 genes differed and 1,800 approximately in common. But because the genome is a little bit larger, again, it's about 18% of the loci that differ on average between any two strains. And this is the same type of dendrogram we saw for H. influenza, showing the amount of gene sharing. It was very interesting that there's this one set that are much closer together, and later on I may talk a little bit about this. Now, another useful thing that you can do is with the supergenome model is you can predict population, how many strains you have to sequence in order to get different coverage levels of the supergenome. So if you want to look at what we call non-rare genes, that is genes that are present in 10%, at least 10% of the strains, in order to get 90% coverage of the supergenome, we only have to sequence 11 strains. If we want to get 99% coverage of the non-rare genes, we have to sequence 33 genomes. But if we want to get all gene frequencies, even just to get 90% coverage, we have to sequence 142 genomes, excuse me. And so also using the supergenome model, we can see that less than half of the genome, the supergenome is core, even just looking at the non-rare genes. And if we include the rare genes, only about a quarter of the species supergenome is core. A rather revolutionary thought if somebody had told you this 10 years ago. And again, like with Haemophilus influenza, we see huge numbers of indels. And all I want to do again is just show the average. The average number of insertions and deletions for all of these strains compared to all these other strains, again, is over 200 as you go around the chromosome. So it doesn't matter whether you're looking at gram negatives or you're looking at gram positives, you see the same phenomenon. And this just summarizes that slide. And again, for another pathogen, Staphylococcus aureus, we see the same thing. The number of distributed genes per genome ranges from 317 to 628. And the core genome and the supergenome, again, the average number of gene differences, again, between 350 and 400. And then just the dendrogram showing the amount of gene differences. We were not expecting to see the high level of gene differences with Staph aureus that we did with Haemophilus influenza and Streptococcus pneumoniae, and that's why we did this. And I'm showing you this data not to bore you, because Staphylococcus aureus does not have the ability, it's not competent, and it doesn't have transformation. And so we were expecting less variability, but in fact we see almost the same level of variability. So it's obviously using other mechanisms for horizontal gene transfer. So what I want to leave you with is that this is a universal phenomenon of this distributed genome hypothesis, regardless of whether we know the mechanisms that the organisms are using for transfer of DNA. And this just shows the relationship. It's a new method we call neighbor grouping for showing the relationship of the various strains based on distributed gene possessions. So now what I'm going to do is talk about something perhaps maybe a little bit more clinically relevant. Comparative genomic analysis of serial pneumococcal isolates. Insights into their supergenome evolution and virulence. And what we did here is we took a single patient who had chronic respiratory infections and chronic middle ear disease, and over the course of 7 months had 13 isolates, serial isolates from this individual of pneumococcus, and we sequenced most of the strains that we had available to us and looked at the amount of evolution within these strains during the actual infectious process in a single child. So what we did is we applied whole genome sequencing to strains recovered from the same patient to investigate the evolution in a single individual in vivo. And so what this slide shows, this is the various visits across the top here, so from 1 to 13. And these are the visits where they actually recovered a pneumococcal isolate. There were a few visits where they didn't. And the yellow ones are ones that we sequenced. And I'm sorry this bar slipped here a little bit. What it was supposed to be doing was going over this row here, what's called MLST. And MLST is a way of DNA fingerprinting, if you will, strains. So what we can see is there's two basic MLST types. So there's a bunch of 13s, and there's also two 2011s. We also see that there's two different serotypes here for pneumococcus. There's a bunch of 14s, and there are several that are non-typable. But what we see is that the MLST and the serotype doesn't always match up. So sometimes we see a 13 with a non-typable, sometimes we see a 13 MLST with a 14. So this suggested to us that there was probably horizontal gene transfer going on between these different strains, and that's why we selected this patient and their strains to look at in detail. And so this, you've already seen this. This is just showing the different phenotypes of these different pneumococcal strains from this individual. I used this same example earlier. This is just an outline of the method. We've pretty much gone over this, so I'll skip over this slide. And what we found was when we sequenced those 6 strains, that we had actually 4 different genomes. These two, which were here, were identical. So the one that we isolated at visit 1 and visit 10 were identical, and the ones that we isolated on visits 12 and 13 were identical. But these were different from this, and this was different from these, and these were different from this one. And so what we have here is just the serotypes, the number of coding sequences, the number of distributed genes relative just to this group of strains, and the number of core clusters. So the first thing I want to do is show you using this neighbor grouping method that this group of strains that from this individual here are fairly closely clustered together compared to the whole pneumococcal world. So that they kind of make a group, which kind of fits with our concept that you've got evolution occurring in situ here, and so that's why you have this cluster of related strains. So what we find out is that there's a total of 51 distributed genes from this group. And what we found when we started looking for insertions and deletions around the genome from these various strains is that there were a total, I'm sorry about the background of this, there were a total of 19 indels different between these strains, or insertions and deletions of genes around the genome. And there were 9 of them that contained horizontally transferred distributed genes, and 10 which just contained transfers of core genes that we could pick up by allelic differences. And so what this slide is just simply showing is just the number of genes that are being transferred during these processes. So each color here represents a different insertion or deletional event. And whether the gene represents a distributed gene here, we say contingency, or whether it's just based on differences in SNPs. And so these slides are just to show you, to give you an idea, how many genes are being moved back and forth between these strains during an infection. So each one of these lines is a different gene. And I do this not to have you look at the individual genes, but just to get a sense of the amount of horizontal gene transfer which is occurring. And then this is just an analysis, a SNP analysis, where we're looking at the regions of, we're looking at if you have 2 distributed genes that are moving and there's genes in between them, is it likely that the genes are moving in block? And so what we see is that if we do a SNP analysis for regions that we think are moving, even the core genes, we see very high levels of SNPs versus regions that aren't moving. And this is kind of a technical point, and if anybody's interested, I can go over it more later. And so this is what we think in summary is actually happening in this patient. This was our mother strain, if you will, which we could found on visits 1 and 10. And the strain from visit 4 donated 3 regions of its genome to form the strain we find at visit 6. Then the strain from visit 4 gives another donation to this daughter strain, and there's yet another donation from an unknown strain that we did not happen to isolate, which then produced the strains we found at visits 12 and 13. So these are the essentially great-great-granddaughter strains here at this point. And what I want to emphasize is that in this 7-month period, that over 7.5% of the genome from the visit 1 to the visit 12 and 13 strains has been replaced. So this is during an active chronic infectious process. So this is kind of proof, if you will, that the distributed genome hypothesis doesn't just hold at some global abstract level, but that it holds in an individual patient during an actual infectious process. And now the next few slides, I'm going to show you some other species, but what I want to leave you with is that the distributed genome hypothesis holds for every bacterial species examined, and not just known pathogens. All bacterial species possess a supergenome. So here we have the results from, I think, something like 24 different bacterial species. Here's Borrelia burgdorferi, causes Lyme disease. Gardnerella vaginalis Campylobacter jejuni causes food poisoning all around the world. Haemophilus influenza, Francisella tularensis, everybody's favorite biological warfare agent, causative agents of strep throat, staph, Listeria monocytogenes, another foodborne pathogen, botulism, Clostridium difficile, gangrene, TB, Salmonella, Shigella, E. coli, Bacillus anthracis, Burkholderia malari, and just the number of strains that we've sequenced and analyzed, the gram status, whether they're gram negative, gram positive, indeterminate, the GC content, it doesn't matter what the GC content is, whether it's low, whether it's high, the size of the genome, it doesn't matter whether it's tiny, whether it's 1 megabase or 10 megabases. And then we look at the percent of the genome that's core. And it varies from a low of 58% for Gardnerella vaginalis to a high of 92% for Mycobacterium tuberculosis. And then this is the number of distributed genes that we see and the percent of the genome that's distributed for each of these species. So with the exception of Mycobacterium tuberculosis and Bacillus anthracis, we see that the core genome on average is in the range of 60 to 80%, with an overall average, even including those, of about 77%. So looking at all bacterial species that we've examined, roughly a quarter of the genes in every genome are non-core or distributed. I just have pulled out a couple examples to look at in a little bit more detail here. So this is Gardnerella vaginalis. And what we see is it has the smallest core genome on a percentage basis, only 58%. And this is extraordinarily striking, we found, because not only is it have the smallest core genome on a percentage basis, the genome itself is very small. It's only about 1.5 megabases. And so that the core genome for this species is only 727 genes. Every strain varies from every other strain, on average by over 500 genes. So it really starts to make you question, how do we define a species? And then here's the other extreme. This is TB, and here we see that the core genome in this case is 92%. And it's believed that one of the reasons the core genome here is so small is that the evolutionary microbiologists tell us that TB is actually a very, very new species, very, very recently evolved. So I think at this point we can conclude that the distributed genome hypothesis holds for all bacteria examined. The core genome in all species are smaller than the distributed genomes, with the exception of those two examples that I showed. Thus, the supergenomes are modeled to be multiple times the size of the core genome, and the observed genomic plasticity provides a plausible means to understand the phenotypic diversity among clinical strains. Now, you can also use the core genome as a tool to determine species boundaries. Some current species boundaries are incorrect, and some species include strains which do not belong. And I just have a couple examples for this. So we think of Haemophilus influenza and Haemophilus hemolyticus, both organisms that reside only in the human nasopharynx, as two different species. But here again we see our classic core and distributed core and supergenomes for Haemophilus influenza. And the Haemophilus influenza additions stop at this blue line. And the next two strains that we add are actually Haemophilus hemolyticus. And what we find is they really don't make almost no difference in the size of the core genome. The supergenome goes up a little bit. And we've now sequenced two more Haemophilus hemolyticus strains. And again, this doesn't change. And so as we start looking at this, what we realize actually is that Haemophilus hemolyticus is actually the species, and Haemophilus influenza is a very successful clade of Haemophilus hemolyticus, which tends to be more pathogenic. If we add other pastoral ACEs, other related strains, we see very rapidly that the core genome drops very substantially in size. It halves here, and the supergenome just shoots off. So we know that these other ones are not part of the same species. And we see very similar things with the pneumococcal. So here's just the actual numbers. So if we look at Haemophilus by itself, the core genome is 1,446. If we add in Haemophilus hemolyticus, the number of core genomes only decreases a small amount. The core genome size is still 364. But if we add another strain from another species, Haemophilus, I think it was somnus, suddenly it halves. It drops to 700. So there is, if you will, a genus core, as well as a species core. But I would argue that Haemophilus hemolyticus and Haemophilus influenza are really the same species. And this again, is just one of our dendrograms. So these are all the Haemophilus influenza. These are the two hemolyticus up here. And then these are the other Pasteuracea way out here in terms of core genome differences. Okay, so this is, I'm going to show you the same thing with pneumococcus, but I'm going to jump to the next slide. So just like we think that Haemophilus influenza is a successful clade of of hemolyticus, we now have very similar data to show that Streptococcus pneumoniae is a very successful clade of Streptococcus mitis. So this is the Streptococcus pneumoniae core genome and supergenome. We add 2 strains of Streptococcus mitis, and we see no difference in the core genome, very little difference, a little bit of increase in the supergenome, as you might expect. But if we add other Streptococcal species, we get a very rapid decrease in the size of the core genome. So again, it looks as if some of these pathogens, human pathogens that we're seeing, are fairly recently evolved. They're maybe in the going through speciation, but we're kind of catching them in the act, if you will. And then this is the dendrogram again for that set of data. I think I'm going to skip over this and just look at here. If you look at the core genome size for Streptococcus pneumoniae by itself, it's 1,700. If we look at it where we add in 4 other species in addition to mitis, it drops to 1,100. So just like with the Haemophilus, there's a genus level core genome as well. In this case for Streptococcus, about 1,100 genes. So you can use the core genome and the supergenome, for identifying species and genus level relationships. And then just one more example of this. So here's Bacillus anthracis, the other organism which shows very low levels of genomic plasticity compared to most of the species. 92% of its genes are core. You're very, very close together. Now, if we look at Bacillus cereus, which people knew to be fairly closely related, we see its core genome on average is 67%. So that was kind of a puzzle. So what we did is we threw together all the genomes for cereus and anthracis. It really didn't make much difference. Core genome for anthracis and cereus together is 65%. So adding in the anthracis really didn't make much difference. So again, we would argue that Bacillus anthracis is, again, just a successful recent clade that has evolved from B. cereus. And if we look again at the dendrograms, this is the B. cereus down here with all the differentiation, and all these are clustered tightly together. Is just this one successful clade of B. cereus that we identify as anthracis. We also can use this technology to show when strains have been misclassified. So we just went to the literature and looked at at Clostridium botulinum. And this was work that was done with Barry Hall. And what Barry found was that if he looked at all the strains that were called Clostridium botulinum, the core genome was only 14%. It just didn't make any sense. And you can see that if you group these, there are these 3 strains that are just these enormous outliers here. And so if you pull those 3 strains out, then you get a much more standard core genome size of about 80%. And what it turns out is those are completely different genus that had happened to pick up the toxin genes for botulinum. And based on that, they've been classified as Clostridium botulinum, but they weren't even in the same genus. So, conclusions. Bacterial plurality explains persistence during chronic infections. There's generation of phenotypic heterogeneity, metabolic heterogeneity, and genetic heterogeneity. The distributed genome hypothesis provides a mechanism for generating genetic heterogeneity and genomic plasticity. The existence of a species-level supergenome, polyclonal infections, and horizontal gene transfer mechanisms among pathogens serves as a population-level virulence factor by producing clouds of related strains which confuse and overwhelm the host immune system. Core genome studies can help with species definitions. Thank you.